Hi everyone, welcome back to my channel, and today we are going to learn about fatty acid metabolism. First of all, let's know about lipids. Lipids are the heterogeneous organic substances that are made up of hydrogen and carbon, and we all know that it is not soluble in water and it is soluble in alcohol and ether. We can simply classify it into simple lipids and compound lipids, and we can further classify these compound lipids into phospholipids and glycolipids. Now let's learn about fatty acid. Fatty acids are the simplest form of these lipids, and we can classify them on different bases. First of all, let's divide them in the basis of their length. One will be the even chain fatty acid. Even chain fatty acids have even number of carbons in their group. So example is palmitic acid. Another will be the odd chain fatty acid. There it will have the odd number of carbons. Example is propionic acid. Another is short chain fatty acid. This contains less than 6 carbon atoms. Example is propionic acid, which have 3 carbon atoms. Another will be the medium chain carbon fatty acids, where it have 8 to 14 carbon atoms. And last, there will be the long chain fatty acid, where it will have 16 to 24 carbon atoms. An example is palmitic acid. Another very important classification of fatty acid is on the basis of saturation of hydrocarbon chain. So we can divide them into two main headings. One will be the saturated fatty acids, and another will be the unsaturated fatty acids. So if the hydrocarbon contains only the single bond, there is no any double bond, then we called it as a saturated fatty acid. Example are palmitic acid, stearic acid, and another is the unsaturated fatty acid, where it contains one or more than one double bond. So we can again classify them in case of, we all know about unsaturated fatty acid, it will have a double bond. So if it has one double bond or more than two double bond, so in that case also we can divide polyunsaturated fatty acid and monounsaturated fatty acid. It will have the one double bond, monounsaturated fatty acid, and in case of poly we have more than one double bond. So again, this in case of polyunsaturated fatty acid, we can divide them into two forms. One will be the trans form and another will be the cis form. Cis form is the natural fat which is available, and trans form is the fat which is available after food processing. So it is not good for health. Trans fats are obviously not good for health. So we should know the main two PUFAs that is very important. That is the linolenic acid and linoleic acid. These are the essential fatty acids. Essential fatty acid means they are not synthesized by the body, so this should be supplied through the diet. And what are the functions of these PUFAs are they can transport of cholesterol, synthesis of eicosanoids required for the membrane structures and functions, and another will be the transport formation of the lipoproteins and prevention from the fatty liver. So this is the basic about lipids and classification of fatty acids. Now let's learn about lipoproteins. Lipoproteins are the complex that are formed from lipids plus proteins. So they are very important, and we can classify these lipoproteins mainly into four headings. First of all, one will be the chylomicrons. These chylomicrons are first of all, let's know the name of these lipoproteins. Very low density lipoproteins, low density lipoproteins, and high density lipoproteins. Okay, first of all, let's know about these chylomicrons. Chylomicrons are synthesized in intestine and they transport the exogenous triglycerides to various tissue. Exogenous triglycerides mean the triglycerides which we consume through our diet. So, and their another work is also to activate the lipoprotein lipase. So, what does this lipoprotein lipase does? It converts the triglycerides into free fatty acids. Another is very low density lipoproteins. They are produced in liver and intestine and they are responsible for transport of triglycerides from liver to tissues. From liver to tissues. Another is low density lipoproteins and they are formed from very low density lipoproteins and their work is to transport the cholesterol from liver to other tissues. Cholesterol from liver to other tissues, so it is bad for our health. And another is high density lipoproteins. They are synthesized in liver and their main work is to transport this cholesterol to liver from different various tissues to liver. So if there is any atherosclerosis in blood vessels, also if there is any kind of lipids accumulating in these blood vessels specifically, then they will carry this to the liver. So this is the basic about lipoproteins. Now let's go on our main topic, main heading today. Today we are going to learn about fatty acid metabolism and there are mainly three important topics divided. One is the fatty acid synthesis, fatty acid storage and beta oxidation. First of all, let's go and learn about this synthesis process. Okay, how these fatty acids are synthesized. I have a diagram here so that we can learn it very easily. So, fatty acid metabolism. So, how it is synthesis? So, whenever there is citrate level increased, for example, we have a lot of energy, we have a lot of ATPs. So, what does this ATP will do is, if there is increase in ATP, then it will inhibit one enzyme and that enzyme is isocitrate dehydrogenase. Isocitrate dehydrogenase. So what will happen is if this isocitrate dehydrogenase is inhibited, then there will be more amount of citrate. Now one very important thing what we have to know is this acetyl CoA can't cross the membrane, it can't directly come into a cytoplasm. So there is one special kind of thing, that is the citrate shuttle, through which this citrate will come from mitochondria to cytoplasm. So now this citrate will convert into acetyl CoA and there will be the enzyme ATP citrate lyase, through which this acetyl CoA will form. Now this acetyl CoA will be converted into malonyl CoA through acetyl CoA carboxylase, and here will be one carbon dioxide molecule and here will be biotin as a cofactor and this malonyl CoA will be formed. This malonyl CoA will ultimately form the palmitate, which is the product we want in this fatty acid synthesis, through which enzyme fatty acid synthase. This malonyl CoA have another very important function, is it inhibits the beta oxidation procedure. Okay, now let's go about the fatty acid storage. So these triglycerides are the fuel reservoirs of our body, especially for this lipids. So whenever we have more lipids, they are stored in our adipose tissues in the form of triacylglycerides, and this triacylglyceride can be broken down into fatty acids and glycerol through this one enzyme, that is the HSL. It is okay, I'll write it here, hormone sensitive triacyl glycerol lipase, and this enzyme is activated by glucagon, and it is also activated when the insulin level is very low, and it can convert it into fatty acids and glycerol. Now let's go in very important topic, that is the beta oxidation. So in this beta oxidation, what happens is we have fatty acid. Now another very important, we have already discussed about short chain fatty acids, medium chain fatty acids, and long chain fatty acids. So this short chain and medium chain fatty acids can diffuse directly into the mitochondria for beta oxidation. So it does not need carnitine subtle. This carnitine subtle is a specialized form which is specially needed for the long chain fatty acids, because the small chain and medium chain can go directly into the mitochondria, but the long chain fatty acids can't go directly into the mitochondria. So what happens is that this fatty acid is converted into fatty acyl CoA through an enzyme, that is fatty acyl CoA synthetase. But we have to know it is only applied in case of long chain fatty acid, so its enzyme name is also long chain fatty acyl CoA synthetase. Now this fatty acyl CoA can go in this membrane. Okay, this is cytoplasm and this is the mitochondria. Now fatty acyl CoA will go into the carnitine subtle. What happens is there is one enzyme, carnitine palmitoyl transferase 1, and with the help of this carnitine it will convert into acyl carnitine. Now this acyl carnitine can go into the mitochondria, can diffuse into mitochondria, and here this acyl carnitine will get converted into fatty acyl CoA through the enzyme carnitine palmitoyl transferase 2. So what you have to know is this fatty acyl CoA have to come from mitochondria, from cytoplasm to mitochondria, and for this all process happens what we call it as a carnitine subtle. Now this fatty acyl CoA will convert it into acetyl CoA. Okay, now we can look this. Now fatty acyl CoA is converted into acetyl CoA through acyl CoA dehydrogenase, and this acetyl CoA can either go in the formation of ketone bodies or it can go in the TCA cycle for the energy. Another very important topic is odd chain fatty acids. So what happens is in case of even chain fatty acids, the process of beta oxidations happen normally. But in case of odd chain fatty acids, beta oxidation will proceed till 3-carbon structure. Till 3-carbon structure, the beta oxidation process will happen. But when it will reach to a 3-carbon structure, that is the propionic acid, then it will convert it into methylmalonyl CoA through the enzyme propionyl CoA carboxylase, and the cofactor needed here is biotin, and this methylmalonyl CoA will convert into succinyl CoA through the enzyme methylmalonyl CoA mutase, and the cofactor is vitamin B12. Now this succinyl CoA can make glucose through gluconeogenesis. So there is one exception to our human body cells, is fatty acids can also make glucose, but only the odd chain fatty acids can make glucose. Otherwise the even chain fatty acids will only make energy through the TCA cycle after the formation of the acetyl CoA. So this is very important thing to know. Another important thing is in this process of beta oxidation, it will produce 3 important things. That one will be the acetyl CoA, which will enter to the TCA cycle for the production of energy, and another will be NADH and FADH2. They both will enter to the electron transport chain. Now this fatty acid oxidation process occurs in liver, muscles, adipose tissue, but not in case of RBC. Not in case of RBC and brain, because we all know that there is no mitochondria in RBC, and so that's why it won't occur in RBC. And another thing, it won't occur in brain also because these fatty acids can't cross this blood-brain barrier. So they can't, the oxidation process won't happen there. And another important thing that we have to know is, while reading this beta oxidation, this carnitine, we have already know about this carnitine subtle. This carnitine comes from our diet. So this is very important one thing. And another important thing is there is use of NADH, NADPH in the process formation of mannoline CoA to this fatty acid, and this NADPH comes from the HMP stunt. This NADPH will come from the N. Okay, this is all for today. I hope you guys enjoyed this, and we will meet in next video.